Hello everyone, welcome to E-Classes by Dr.Athira. This video on human embryology is a brief narration of the process of human development in a highly simplified capsule form and an introduction to development of phase. So first of all, let me tell you what is the process of fertilization or formation of a zygote. As you all know, one human cell contains 46 chromosomes, out of which 44 are autosomes and 2 are sex chromosomes. And on this 44 autosomes or 22 pairs of autosomes, one chromosome is derived from mother and other from father. So let me tell you what are gametes or germ cells. Germ cells, there are two, male germ cell known as spermatozoa and female germ cell known as ova. Both of these contain only 23 chromosomes, that is half the number of a human cell. And they are formed by a cell division known as meiosis. What is meiosis? Meiosis is a type of cell division that produces daughter cells with half the number of chromosomes, that is 23 numbers of chromosomes, and it is by meiosis a spermatozoa and an ova are formed, or germ cells or gametes are formed. It is by the fusion of gametes one zygote is formed, or this process is known as fertilization. So when spermatozoa and ova with only 23 chromosomes in each join, the original chromosomal number, which is 46, is retained in a zygote. So a zygote contains, like any other human cell, it contains only 46 chromosomes, and it is from the zygote or from the single cell whole human body stems from. So as I said, formation of zygote by fertilization marks the commencement of formation of a new individual, that is all cells in human body stems from a zygote. So zygote is a single cell with 46 chromosomes which undergoes proliferation by a type of cell division known as mitosis. The mitotic cell division is a type of cell division where the number of chromosomes are 46 even in the daughter cell. That means parent cell will have 46 chromosomes. Two formed daughter cells also will have 46 chromosomes in each. So by mitotic cell division, the zygote proliferates to form a ball of cells known as morula. And this proliferative phase continues till all three germ layers are formed. The germ layers include ectoderm, endoderm and mesoderm, and it is from these three germ layers all the structures of human body are formed. So let's see how a morula turns into a triploblastic or three-layered embryo. Three-layered, I mean to say three germ-layered embryo. As you can see here, this ball of cell is known as morula. Fluid seeps or fills in between these cells, forming a cavity like this. This cavity is known as blastocyst. There are two population of cells, as you can see here in blastocyst. One population of cell lining this cavity, which is known as trophoblast, and one population of cell seen as a cluster at one pole of this cavity, which is known as embryoblast. Trophoblast forms the covering or lining of this cavity, which is known as primary yolk sac, and it is from embryoblast whole embryo or human body is going to be developed. Or this is, you can consider the structure as a embryo proper. The trophoblast cells are associated with implantation of embryo in the uterine wall, and later it's also associated with formation of placenta. And the embryoblast, as I said to you, they form the embryo proper. And it is from these cluster of cells the germ layers derive from. And the formation of blastocyst is complete by first 1 week of gestation, and from 8th day of gestation onwards, the cells of embryoblast starts organizing to form a two cell layered disc known as bilaminar disk. two cell layers are two germ layers, which are ectoderm and endoderm. So the cells of embryoblast reorganize to form these two layers known as ectoderm and endoderm. This part is magnified here. You can see the ectodermal layer situated dorsally or above, and the endodermal layer located ventrally or below. And this ectodermal layer is forming the floor of this cavity known as amniotic cavity, and the endodermal layer is forming the roof of this cavity known as secondary yolk sac. So by this second week of gestation, there is formation of this bilaminar disc, that is the ectoderm and endoderm in the embryo. So this stage we can call it as a bilaminar disc embryo. So this image shows the ectoderm and endoderm germ layers. Now itself, I want all of you to imagine three-dimensionally to understand the structure as not just two layers of cells, but as two planes of cells, as you can see here in this image. Here you can see one germ cell layer above, which is known as ectoderm, and the cell layer or germ layer present ventrally or below, known as endoderm. And by formation of bilaminar disc, one more important change happens, or one more important point to be noted is the long axis of the embryo is established. That is head to tail axis of the embryo. Here this is the head end or rostral end, and this is the tail or caudal end. Here in this cut section also you can appreciate the bilaminar disc two-dimensionally with ectoderm and endoderm, and amniotic cavity above and secondary yolk sac below, roofed by endoderm. One more structure need to be discussed here, or one more structure develops here, which is known as procoron plate. You can see the cells of endoderm and ectoderm slightly enlarged at this head or rostral end. At this area, there is firm union between these two germ layers, which is known as procoron plate. So procoron plate is firm union between ectoderm and endoderm at this point in the head or rostral end of embryo, as you can see in these two images. And this procoron plate is the future buccopharyngeal membrane. So all these developmental events are complete by 2 weeks, and after 2 weeks, as bilaminar disc formation is complete and procoron plate is formed, there are events that cause the formation of mesoderm and neural tube. So now let's see what are the developmental events that take place after 2 weeks of intrauterine life. So by 3rd week of intrauterine life, there is a structure forming known as notochord. Let's see what is this. There is formation of groove-like structure along the midline of the embryo, that means along the long axis or head-tail axis of embryo within the ectodermal germ layer. This groove-like structure is known as primitive streak. You can see here the development of primitive streak starting at the caudal end or tail end of the embryo, and towards the head end it ends in a depression known as primitive node. At primitive node, ectodermal cells proliferate and pushes themselves towards the procoidal plate. That means ectodermal cells from this primitive node forms a column of cells, proliferate and forms a column of cells that extends towards the procoidal plate. This solid column of cells later canalizes to form a structure known as notochord. The notochord plays an organizational role in development of nervous system, and in the later developmental time it forms the vertebrae in humans. So as the notochord formation is complete, now let's see how is the third germ layer or mesoderm formed. As I said, procoidal plate is the area where there is firm union between the ectoderm and endoderm in the head end or rostral end of the embryo. Ectodermal cells continue to proliferate along the sides of procoidal plate, and newly formed cells spread between the ectoderm and endoderm, that is newly proliferated cells, which forms the third germ layer that is known as mesoderm. Thus, ectoderm and endoderm are completely separated by mesoderm and notochord except at the region of Procoron Plate. So, at the region of Procoron Plate, there is no formation of mesoderm, that is a firm union between the endoderm and ectoderm. So, you can see here the cells of ectoderm proliferate and fills in between the area of ectoderm and endoderm, forming the third germ layer, mesoderm, in between the first two germ layers, that is ectoderm and endoderm. So now if you see the structure of embryo, it is a trilaminar disc or three-layered embryo with formation of ectoderm above, endoderm below and mesoderm in between the area, and there is notochord in the center or along the midline. So all three germ layers are formed by end of third week of intrauterine life, and next 3 to 4 weeks there is development of major tissues and organs from this three-layered embryo. So these are the major developmental events that take place in next 3 to 4 weeks. That is differentiation of major tissues and organs including the hidden phase and tissues responsible for teeth development, differentiation of neural tube from ectoderm, differentiation of neural crest cells, and differentiation of the mesoderm, that is the third germ layer formed. So let's see each of these events in detail now. First of all, the formation of neural tube, which is the primitive nervous system. So at the head end or rostral end of the embryo, the ectodermal cells along the midline shows a slight thickening, that is by proliferation of cells, which is known as a neural plate. So in this diagram, the central part of this ectoderm you can see slightly enlarged, which is known as neural plate. On either side of the neural plate, these sides of the neural plate, there is slight elevation as you can see in this image. These elevated areas are known as neural fold, and automatically the central area becomes like a groove or depression, which is known as neural groove. And later what happens is the sides of this neural fold come and join with each other or fuse with each other, forming a tube that is known as neural tube. So thus neural tube is formed, which is the primitive nervous system or nervous structure from which the brain and spinal cord developed from. In the head region, the neural tube undergoes massive expansion and forms forebrain, midbrain and hindbrain. The hindbrain is again divided into 8 segmented blocks known as rhombomeres. Let's see about a specialized group of cells known as neural crest cells. These cells are present along the lateral margins of this neural tube in the ectoderm, and they are pluripotent cells and they have the property of migration and extensive differentiation to form various structures of head and trunk. To the head and first pharyngeal arch region, they originate from the neural tube, that is the ectoderm on either side of the neural tube in the region of midbrain and first 2 rhombomeres of hindbrain. And these are the various derivatives of neural crest cells in the head region. They form cranial sensory ganglia, connective tissue of head and all the structures of tooth except enamel. All the connective tissue of head and neck is formed from neural crest cell except enamel of the tooth. So in the whole head and neck region, the only structure which is not a derivative of neural crest cell will be enamel. Tooth enamel is formed purely from ectoderm germ layer. Connective tissue elsewhere is derived from mesoderm and is known as mesenchyme. But in the head and neck, since there is a role of neuroectoderm or neural crest cells, the connective tissue is termed as ectomesenchyme. And in the trunk region, they differentiate to form various structures like neural cells, Schwann cells, melanocytes and sensory ganglions. So the another event that take place during this developmental time is differentiation of mesoderm. Mesoderm on either side of the neural tube is getting segmented into various blocks like somites, paraaxial mesoderm, etc. but I'm not going into detail of it in this video. But I will tell one thing, that it gives rise to muscles and connective tissue, that is mesenchyme beneath the skin. So from end of third week to end of fourth week, that is from 21st day to end of fourth week of gestation, very important change happens to the embryo. As you can see here, the embryo, head end, tail end, and the head tail axis, there is folding of the structure. Folding take place in the head region as well as tail region in this plane, that is head tail axis, as well as in one more plane, that is the lateral plane, as you can see here. So embryo gets folded in head tail axis and in the lateral plane to form a structure like this. So hereafter in the development, the embryo will be having this particular form. This folding of embryo is critical in development of primitive oral cavity or stomodium. You can see the folded embryo here. There is a gap between the primitive head region and the trunk region. This gap is known as stomodium or primitive oral cavity. Here you can see the developing gut or gastrointestinal tract. The primitive oral cavity is separated from the gut in the initial stages by a membrane known as buccopharyngeal membrane, which is nothing but the procaudal plate. We had discussed in the region of procaudal plate, there is firm union of ectoderm and endoderm. So procaudal plate is the one giving rise to buccopharyngeal membrane, which is a separation between the primitive oral cavity or stomodium and the primitive foregut. So this video is a briefing of general embryology and an introduction to development of face. In my upcoming videos, I will be discussing development of various structures of face. Thank you for listening.